We're going to start Unit 7A, Part 2, with spermatogenesis. Spermatogenesis is the process of production of sperm in the seminiferous tubules of the testes. Every cell in the body, and this applies to male and females, every cell in the body has 23 pairs of chromosomes. The 23 chromosomes are called homologous chromosomes because you have one copy of each one from your mother and one copy of each chromosome from your father. So if we look at, let's say, chromosome 16, you have one copy of chromosome 16 from your mother and one copy from your father. These cells throughout your body all contain those two copies for each chromosome, and therefore are considered diploid. They have a 2N chromosome number. Let's get that up here. Yeah, 2N chromosome number. 23 pairs of homologous chromosomes, one maternal in origin, one copy is maternal in origin, and one copy is paternal. However, in sexually reproductive cells, you only have one copy of each of the chromosomes. So you have 23 chromosomes as opposed to pairs of chromosomes. You have 23 chromosomes, and this is the haploid number of chromosomes, or N chromosomal number. When these cells get together by fertilization of the female egg by the male sperm, then it gets back to the normal diploid 2N number of or chromosomal content. Okay, now this reduction or halving of chromosomes in spermatogenesis is achieved by a process called meiosis. It's reductive division. This diagram shows the process, and it shows it in two ways. One is at the individual cell level on the left-hand side, with emphasis on the chromosome count. So whether it's diploid, 2N, or haploid, single N. And on the right-hand side, anatomically, going from the outer edge of the seminiferous tubule, this is a slice of a cross-section through the, you have a cross-section through the seminiferous tubule. So if you have a cross-section like this, what is represented here is just a, strange. It's just a slice of that pie. Okay, here and here. That's like a slice of this cross-section of seminiferous tubule. So what's shown here is that the cells at the outer perimeter, these cells that are found at the outer perimeter, this is the stem cell, and there's stem cells all along the outer edge. They're called spermatogonium, and they are the cells that will produce sperm cells within the seminiferous tubule. Here at the opposite extreme is inside the space, the central space, where you have release of the mature or almost mature sperm cells, spermatozoa. They still have a few things to go through to be motile, but basically they're there and they're fully formed. And in between, we have all the processes just here, illustrated here, that are occurring, and illustrated here that are on the other side that are occurring in terms of the chromosomal count. So what does happen, and all this is described in detail in the following slides. I'm just running through it here once, and then we'll go through it on the individual slides and descriptions. You have the spermatogonium cell, which is the stem cell, and it can divide, and it undergoes mitosis. So there's the spermatogonium cell with a diploid cell, diploid chromosome count. It divides by mitosis into two daughter cells. One daughter cell, the type A spermatogonium, will replace the cell that divided. So you have a replacement of stem cells, otherwise you would run out of stem cells. And the second one is the type B spermatogonium, which is going to go on to differentiate and divide into forming sperm cells. So we have the type A spermatogonium and the type B spermatogonium. Now all of these are still 2n in terms of the chromosome count. What happens next is that there is differentiation into a primary spermatocyte. So from spermatogonium to type B spermatogonium to primary spermatocyte, and this is starting into the process of meiosis. So there is an initial step here, meiosis 1, followed by meiosis 2. And what you have here is that there is a actual doubling of the chromosome count. It's not shown in terms of n, but I'll describe it in a little while later on the slides. But it differentiates into the primary spermatocytes, and then they undergo the reductive division process of meiosis to secondary spermatocytes that are haploid, or N. So there's the secondary ones. And then they further divide into 4 early spermatids. So you go from spermatogonium to type B spermatogonium to primary spermatocyte to secondary spermatocyte to spermatids, which, you know, go through an early phase and then a later phase. They're differentiating so that they then form, they have quite a few changes, but the major one is that they form a tail, or better called flagella. So each of these is forming a flagella, so that they are the only flagellated cell in the body, the only cells with flagella. So these are mature sperm, or also called spermatozoa. There is a process that's important in spermatogenesis, production of sperm cells, that occurs at the last phase, and that is called spermiogenesis. So spermiogenesis is the last part of spermatogenesis. Spermiogenesis involves this maturation and differentiation into this cell type with a flagella. Okay, let's go through. Oh yes, there is one other important thing I wanted to point out to you, and I'll describe that later in the slides, but for now I just want to point out to you that there is another cell here that may not be obvious to you. There's two other cells that I'll outline one of them. First of all, here is the, no, that's not going to do it. Here is the nucleus of that cell right there. And now I'll outline the whole cell body. It is here, the entire cell, and I already highlighted the nucleus, so now I'm just outlining the cell itself. This is called a sustentacular cell. And what it does is it forms a barrier all around these developing sperm cells, so that the structures or molecules associated with on the surface or inside the sperm cells never actually get into the person's bloodstream. So this individual is, that goes right there, yeah. This individual never sees, the molecules associated with these cells never enter into that person's bloodstream, the person who's produced them's bloodstream, and I'll explain right at the end, I'll explain why that is and why that's important. There is a second one shown here, again surrounding all of these cells so that nothing can enter into the body of the person producing these sperm cells. Okay, so just to point out again, this process involves meiosis. Okay, there is a mitotic division right at the start where the spermatogonium becomes a type A and type B spermatogonium, so that you replenish the stem cells, and the other one, the type B, goes on to producing sperm cells. But there's also a reduction from 2N to N, from diploid to haploid. And the other thing that occurs in meiosis is that there is a introduction of genetic variability. So the sperm cells are not identical to each other. If all the sperm cells were identical to each other, or all eggs that a female produces were identical, then all your children would be identical. And as we know, that doesn't happen. There are actually two divisions, two cell divisions or nuclear divisions occurring during meiosis. It's meiosis 1 and meiosis 2. It does result in a halving of the number of chromosomes from 2N to N, and it also results in 4 daughter cells being produced, and each of the 4 daughter cells are haploid. So you have 4 haploid cells. Okay, 4, I should really write it this way, 4, sorry, let me just get this, 4 cells are produced, spermatozoa or sperm cells, and they have N chromosome count. Okay, now just to help you understand how you can go from one type B spermatogonium to 4 daughter cells, I just wanted to point out to you, and it's important that you understand that the chromosomes actually do replicate just prior to meiosis 1. So you get a doubling or replication, and then goes into meiosis. So if you're starting with a cell that's 2N and it doubles everything, then you have 2 copies, and then if it goes through meiosis 1 and meiosis 2 and reductive division and cell division, you'll end up with 1N in each of the produced so-called daughter sperm cells. Okay. So actually I'll describe that process here, because a lot of people aren't familiar with the fact, a lot of people perhaps are familiar with the fact that, chromosomes are illustrated as being looking like this, usually, that kind of a X pattern. What they don't tell you is that, in fact, there's only a certain time during the cell cycle that you can see chromosomes, and it's only when they are like this that they're visible. Truth of the matter is that this is 2 copies of the same chromosome. This shows 1 and 2. They're separate single chromosomes that just happen to be joined right at the center here. Okay, so they're joined there. Let's call this, for argument's sake, chromosome 3. Okay, so chromosome 3, you have 2 copies. Now, you started with 2N, so that means, of course, that you had the person had a paternal copy and a maternal copy of chromosome 3. And on top of all that, there was a doubling of it. So there's the maternal copy of chromosome 3. Let's get rid of these. So now you know that there's, let's put a little center joining point to remind you that these are 2 copies of chromosome 3. Ooh, I can't put that on there. Let's see, maybe this. Okay, right there, join there. So we have the paternal, 2 copies of chromosome 3, and we have a maternal, 2 copies of chromosome 3 here. Now, during meiosis, and I should point out also that these are called tetrads. If these are chromosome 3, the equivalent also is present for chromosome 1, 2, 4, 5, 6, 7, 8, equivalent, you know, and these tetrads. So 4 copies, 2 copies now of the paternal and 2 copies of the maternal. These these are homologous chromosomes. They're all chromosome 3. 2 are paternal, 2 are maternal. They're homologous to each other. What happens during meiosis, during that first phase of meiosis, meiosis 1, is that they line up next to each other, as shown here in this part of the diagram here. And they line up and they exchange bits of with each other. They exchange the equivalent parts with each other. And so you end up with, and this process is called crossing over. Okay, so here you have this exchange occurring right in that region there. So the end of this part of, we were calling it chromosome 1, the end of the part, the maternal chromosome 1 and the paternal are exchanged with each other. In other words, what you end up with, I'm going to draw over this one, is you end up with this situation. Okay. There. Now what happens as the cells go through meiosis 1 and then divide further into meiosis 2 is you end up with 4 eventually you'll end up with 4 spermatids, or that go on to form 4 sperm or spermatozoa, that each have a copy of chromosome 3. And what you have is one of those sperm cells has a copy of chromosome 3 that looks like this. Another one has a copy that looks like this. Another one will have a copy that looks like this. And then the 4th one will have a copy of chromosome 3 that looks like this. Okay? So this kind of crossing over that occurs during meiosis 1 adds quite a degree of genetic variability into the process. And so all the sperm are not the same. I should also point out to you that when you have that each of the different chromosomes will sort out independently of the other into the maturing differentiating sperm cells. So you may have just as here you have, you know, these 4 types of possible potential types of chromosome 3, you have a similar process going on for 4 and 5 and 6, etc. And it doesn't mean that, you know, I mean, you get a, well, let me draw it for you. Let's say we're talking about chromosome 1, 2, let's keep it simpler, 3, etc., right? 4, 5, 6, etc. So, you know, a paternal copy of chromosome 1 may go into a sperm cell, and then the next chromosome 2 in this sperm cell is maternal in origin, chromosome 3 is maternal in origin, chromosome 4 is paternal in origin. Another sperm that gets produced may have will have a different combination of which origin of chromosomes where they come from, whether it's maternal or paternal, a different pattern. So between random assortment of chromosomes and crossing over, you get tremendously huge variation among all of the sexually reproductive sperm cells that are eventually produced. And yes, they have characteristics that are in you, but they differ from each other also. Okay, let's move on. So this is two processes for genetic variability that are occurring during this meiotic period or meiosis. In meiosis 2, as I said, you have this division. Remember, you did have 2N and the chromosomes doubled and they went through crossing over, and then they were randomly segregated into different cells that were produced, and you'll get 4 sperm cells from the original 1 spermatogonium. Now, this is just a review. Spermatogonium forms spermatocytes. They go on to undergo with meiosis. They form spermatids, and then you get sperm eventually. Okay. Spermatogonia are the outsidemost cells in the seminiferous tubule. They are in contact with an epithelial basement membrane or basal lamina. The process of spermatogenesis only begins at puberty. Okay. And it's at that point that you get type A and type B spermatogonia forming. Type A cells stay at the basement membrane and maintain that germline, maintain the number of stem cells and ability to keep producing. And the type B spermatogonia, type A, type B spermatogonia, they will start moving towards the interior lumen. They push down towards the center of the seminiferous tubule, towards the lumen, and are differentiating and undergoing meiosis. They become first primary spermatocytes, and then they undergo meiosis and form secondary spermatocytes. And those secondary spermatocytes divide further during meiosis 2 and form spermatids. The spermatids are the ones that are closest to the interior round circular space of the tubule, and will differentiate into spermatozoa. Okay, so that's progressive spermatogonium, type B spermatogonium, primary spermatocyte, secondary spermatocytes, spermatids, and finally late spermatids and spermatozoa. And this process involves an initial mitosis here, forming the type A and type B spermatogonium, and then maturation to primary spermatocyte. And remember that the DNA actually doubles so that you get more. It's still called 2N, but it's now looking like that diagram I showed you before. Okay, where one double is maternal, one is paternal. And then meiosis 2, where they divide down and they segregate out, and you get generation of tremendous genetic variability, and then spermatids. Going from early spermatids to late spermatids and spermatozoa is the process of further maturation that is called spermiogenesis. Okay, so spermiogenesis is just this part of the whole entire spermatogenesis process. It's the conversion of spermatids to mature sperm. They are haploid but not motile in the end. After spermiogenesis, they cannot. After spermatogenesis, the spermatids are haploid but not yet motile, completely or they don't have a flagella. Okay, so during spermiogenesis, they will lose some excess cytoplasm and form a tail, becoming sperm or spermatozoa. The sperm itself, each individual sperm has 3 sections: a head, a midpiece, and a tail piece, or not tail piece, tail. The head contains the DNA and is covered at the very tip by a acrosome that forms a helmet-like structure. It contains enzymes. The enzymes are used by the sperm cell to penetrate and enter into the egg if it arrives there. Then many arrive at the same time, actually, and only one will be able to fully penetrate and fertilize the egg. The midpiece contains lots of mitochondria because there's lots of energy that's required for this voyage up the ductular system of the female, of the reproductive tract in the female. Most fertilizations actually occur in the uterine tubes. I think that's the fallopian tubes or uterine tubes. The older name is the fallopian tubes. So they have quite a ways to travel up from the vagina into the uterus and up into the uterine tubes. The tail, of course, is a typical flagella structure. It's the only cell in humans that has a flagella, but it is not really constructed any different than flagella on a protozoal organism, a single-celled organism that lives in a pond. It's very similar in structure. Here you see that a head with the acrosome on the top here. The acrosome, the nucleus is just below that in this purple area here, and this is the mid-piece that's filled full of mitochondria. I just want to, and of course the tail, I just want to point out to you an important point, and that is that none of the mitochondria associated with the sperm cell will enter into the egg when the sperm fertilizes the egg. Now, I'm not sure if you know this, and this is really for your own information rather than I wouldn't test you on it. But mitochondria are not are structures that are not coded for by the DNA in the nucleus. Mitochondria have their own DNA. They are self-replicating organelles within a cell. And because they are self-replicating, have their own DNA, and none of the mitochondria in a fertilized egg come from the sperm, then that means all of the mitochondria come from the female egg, and therefore all the mitochondria once it's fertilized, all the mitochondria in all cells will be maternal in origin. So all the mitochondria in your body in every single cell are essentially the exact same as the mitochondria in your mother and her mother and her mother and your mother's 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 mother. Okay? Yes, there are some changes with time, small mutations here and there, but essentially it's one maternal mitochondrial lineage. Okay, I did tell you that I was going to talk about the sustentacular cells. The old name is Sertoli cells. A lot of structures and cells in the reproductive system and some other systems also in the human body were named after the person who discovered them, but they've moved away from that, which is why fallopian tubes aren't called fallopian tubes anymore. They're uterine tubes. So Sertoli cells are sustentacular cells, the ones that separate the developing sperm cells from the rest of the person's body. Cells extend down through that whole space that the maturing, developing, dividing reproductive cells are forming in. They have a very tight junction between. It is a tight junction, forming a continuous layer separating out the cells. There are two major compartments. The outermost, in general, in the seminiferous tubule, the outermost basal compartment, and the innermost adluminal compartment. The adluminal means towards the lumen, towards the central space. Here, this is in the adluminal compartment. That's where you have the meiotic division of cells during the process of spermatogenesis. Here in the basal compartment, what you have are the 2N spermatogonia and primary spermatocytes. There's a primary spermatocyte, and there's a spermatogonium. There's a spermatogonium. This is the basal compartment here, and this is the adluminal compartment. What you should understand is that once you get into meiosis, and it's down here where meiosis is occurring, right? So all of these cells are produced or undergo meiosis. With meiosis, what you get is cells that don't express non-self molecules or non-self antigens. Non-self in terms of the male, the adult individual who's producing those sperm cells. I remind you that the immune system, as the immune system develops, what you get is the development of tolerance. So during embryonic development and for a very short period after birth, tolerance is being developed, and the immune system no longer reacts or responds to self, only non-self. All the immune system will only react to non-self. Any T cell, and actually also it applies to B cells, any T cell or B cell that has the ability to react to non-self, any T cell or B cell that has the ability to react to self, in other words, has a antigen receptor on its surface that could react to self, is destroyed by clonal deletion. We discussed it just recently when we did the immune system. So you don't react to self, but you do react to non-self. But those structures have to be exposed to the immune system right at the time of embryonic development, fetal development, and early on in just after birth, very early after birth. Sperm are not formed until puberty. And so when the sperm cells are formed, yes, they are self-like, but with meiosis, but at least the spermatogonia are self, but with meiosis, you start getting expression of molecules that are non-self. And so these cells that have undergone meiosis have to be isolated away from the immune system of the adult male individual who's producing those cells. So there are tight junctions that form a blockade or a barrier, and it is called the blood-testis barrier, preventing any sperm antigens from getting into the bloodstream of the person who's producing them. Okay, spermatocytes and spermatids are completely enclosed by those sustentacular cells. They don't only form that blood-testis barrier, they also deliver nutrients. They help move the cells along towards the lumen, the developing cells that are undergoing meiosis. They secrete some fluid to help with transport of the sperm through the seminal vesicles and up, to get them out of the testes into the epididymis. They help dispose of excess cytoplasm sloughed off during spermiogenesis, and they overall can help sustain and regulate the whole entire process of spermatogenesis. And that's the end of Unit 7A, Part 2.